Cool. Okay. So we'll start with question number 1. That's person with mutation and is missing amino acids 250 to 290 on the BRCA1 protein. A little brief background, that is what's located at amino acids 200 to 300 in BRCA1. That's the NLS, which is the nuclear localization sequence. And so we'll start with this question, the consequences to the protein. So obviously the NLS would kind of be affected because the mutation on the amino acids 250 to 290 falls under the range of 200 to 300. So one consequence would be that the NLS is being affected, and for that one, that would affect the BRCA1 protein from getting to the nucleus for DNA repair. And DNA repair occurs in the nucleus, so it's kind of important that it gets there. And so the BRCA1 protein would not be able to get to the nucleus efficiently or entirely. So that's one consequence. Another consequence is, so the P53 and RB proteins that fall under the range of 200 to 300 amino acids, those would kind of be affected or impaired. And that would affect transcription regulation as well as tumor suppression, because that's what the function of the proteins are. So that's that. And then subsequent process that would be eliminated, I would say, is homologous recombination mediated DNA repair, because the BRCA1 protein can't get to the nucleus efficiently to do the DNA repair, so that process can't really occur. And so as a result of that process not really occurring, the damaged DNA just kind of builds up and accumulates because it can't get fixed, has nowhere to go, nothing to do really. So it just kind of builds up, accumulates, and that's pretty bad because that can cause other mutations to occur as well as uncontrolled cell growth, which can lead to bad things. And so, consequence that could happen to the person, I'd say this person has an increased risk of cancer because the damaged DNA just builds up, doesn't really do anything, can't get fixed, just stays there, accumulates. It can cause that uncontrolled cell growth to occur, which can cause cancers to arise. And so that person, I would say, would have an increased risk of cancer. And because the BRCA1 protein is pretty active in ovaries and breast, I'd say the highest, the types of cancer that they'd be most at risk for would be breast cancer and ovarian cancer. Okay, so question number 2, person with mutation in exon 5 of LDLR, and so the function encoded is eliminated. So the brief background for this, the function encoded by exon 5 and LDLR is ligand binding. That falls within that, it's the 2 through 6 is that ligand binding domain. And so consequence to the protein, so the LDLR receptor would not be able to bind to the LDL particles, so they would kind of stay in circulation in the bloodstream and they wouldn't bind to the receptor. Another consequence would be that the receptor could get misfolded due to the mutation, and in that case it would just stay in the endoplasmic reticulum, and I think it would just deteriorate before it could get to the plasma membrane. That's another consequence. So subsequent process that would be affected is receptor-mediated endocytosis, and that would be because the LDLR particles cannot bind to the LDL, or the LDLR receptor cannot bind to the LDL particles. They stay in the bloodstream, stay in circulation. They cannot be brought into the cell because the binding is not occurring. And endocytosis is when the cell brings something in. So if they can't be brought in, endocytosis isn't occurring. So the receptor-mediated endocytosis would not occur. Consequences to the person. I would say this person would have hypercholesterolemia because of all just like the loose LDL particles floating around in the bloodstream. So that those are increased. Those would cause levels of cholesterol in the bloodstream to increase. And so I would say this person has hypercholesterolemia.